I'm really glad to have Dora Hermes visiting today from the frosty Midwest. And just in time to see the daffodils, saw a few daffodils this morning, so spring is finally on its way. Dora completed her PhD in Holland, a place known for its flowers, especially tulips and mother bulbs at the University of Utrecht, where she worked with Nick Ramsey and others. Then she moved to the US, where she did postdoctoral studies at Stanford with Joseph Pavizzi and NYU with John Winauer, and then started her own lab at Mayo Clinic in 2019, where she's now a tenured professor in the Department of Biomedical Engineering. Dora is a world expert in two of the most powerful techniques we have for examining the human brain, old fMRI and intracranial EEG. She's one of the few people who successfully fused those two techniques to really make new discoveries about how human cognitive function work, especially the visual system. And she's also been a real really pushed the envelope in developing new techniques and methods for analyzing data, building new models for linking IEG and fMRI. She was the senior author on a paper that described the BIDS IEG data format, which is a really important step in the community for sharing data and making science more open. She's developed software for localizing electrodes in intracranial EEG patients, which has been really important in our work developing RAID packages that work on similar problems. So it's really easy to see how high impact her work is. It's been cited, I think, more than 4,000 times now, and she has so many dozens of papers on bioRxiv. You get a ping like every day when a new one gets posted. So it's really exciting to see all the stuff that she's working on. And recently, Dora has become enamored of electrical brain stimulation, which is a really powerful technique for measuring how brain activity relates to cognitive functions. And she's going to tell us about some of that new work today. Thanks, Dora.
Thank you so much for having me here. It's really a pleasure to be able to visit, and thank you so much for hosting and for the nice introduction. I'm excited to tell you about some of our electrical stimulation driven neural work on the individual pathways. And before we dig into the electrical stimulation, I want to just talk briefly about the visual aspects of why we're doing this type of work. Because I'm at Mayo Clinic, there's always a clinical relevance. They always want you to see using these things in patients. So I'll first talk about that for a little bit. And so when you look at these pictures, this is actually a picture of my kids about 8 years ago when they were still little and cute. But what I want you to look at are these bars and checkerboard patterns, because those are extremely common. And they can actually surprisingly trigger seizures in patients with pattern sensitivity. If it was larger than this example. And so in pattern sensitive epilepsy, it only affects very few patients. It's often can be easily treatable with medication. But when it can't, like for example, in Dravet syndrome, it can have a large impact on a patient's life. For example, for 2 years, there was a family who reported only going to the playground at night. Because the stripes on were too bright during the day, or people moved to the countryside to avoid like very strong visual stimulation, or they changed the interior of their house. And there's no animal model for pattern-sensitive epilepsy that matches the human phenotype. So we can't necessarily use animal models to understand what's going on in these patients. We have to learn more from human measurements. And differently, there's also different types of epilepsies, different types of focal epilepsies that really affect a small region in what they call in clinical terms the posterior quadrant, so in different visual areas, and that affects about 5 to 10% of patients with focal epilepsies. And so there's different types of diseases that affect these visual pathways, in which we want to understand more about the types of signals that we have in these pathways. And also, how can we modulate those areas with electrical stimulation? How can we use electrical stimulation to see whether those responses, for example, change? So the first thing I would like to talk about today is how can we integrate these human brain measurements to better understand pattern sensitivity? And then how can we use electrical stimulation to influence these visual pathways? So we can learn a lot of different things from different techniques, for example, and we generally compare those on like a scale of spatial resolution and temporal resolution with techniques like EEG, intracranial EEG, or local field potentials, all having a high temporal resolution, but varying on the spatial resolution. And on the other hand, we have fMRI, which has a good spatial resolution, but of course lacks a little bit of temporal resolution. And then we directly compare these measurements and see this one tells us this and this one tells us that. And in some cases, we've learned a lot from these things, like we know a lot of these differences in these different techniques. But in some of these diseases like photosensitive epilepsy, evoked potentials, neural synchronization, and fMRI all increase in amplitude. So it doesn't necessarily tell us what's going on in these cases. And so what I would like to emphasize is an approach where we think about what human brain measurements encode. So I would like to think about how these measurements are driven, in this case, by different stimulus properties. So how, rather than directly comparing them. And then I'm going to focus on this kind of millimeter scale that we capture with intracranial measurements and fMRI measurements. So how can we learn more about this scale of measurements? And what does that tell us compared to the other approach? So let's take this approach and then come back to the question about photosensitive epilepsy. When we look at these types of measurements, we can measure, for example, fMRI and intracranial measurements. In intracranial measurements, these electrodes are placed in many different regions on the cortical surface, sometimes also covering some visual areas. And what we actually need to do in this case is we need to think about the different types of signals that we can extract from these electrodes, because we have field potentials, oscillations, broadband activity, and each of those measurements, we can compare those to fMRI and also draw different conclusions about what we can learn from each of these. And I would specifically like to emphasize, in this case, two different types of signals. First of all, the narrowband gamma oscillations that occur between frequencies between 30 and 80 hertz have often been reported and really reflect an oscillation. So synchrony between areas, have a peak in the power spectrum. This is crucial at around somewhere between 30 and 80 hertz. Are they're thought to reflect like a synchrony or feed-forward communication. However, around later in time, when people started more looking into detail in these intracranial recordings, people also saw that these field potentials also express like a random walk and basically change in a broadband manner across many different frequencies, basically in a 1/f approach where the whole frequency spectrum switches up and down, and it has been correlated with possible input firing rates. Now, when we think about these different signals, what I said is that we want to understand how those are driven by different types of inputs, different types of stimulation. And this has also been referred to as broadband gamma or high frequency broadband. And so when we think about using these linking those to different stimuli, it's a very common approach to use a large grating and put that on the screen. And then we can see on a single trial basis, this is a single trial of an intracranial recording in occipital cortex, is that we see this large evoked potential followed by this synchronous, this gamma oscillation in the raw data. And we can see this sometimes on even a single trial basis. And also when we average across stimuli, we can similarly see that if this is baseline and the dashed line, you can see a broadband increase, but also this strong peak in the signal when patients look at these gratings. However, when they look at noise patterns, we similarly see an evoked response, but then we don't really see anything in a raw signal because it's a random walk. But when we look at the power spectrum, we can see that there is a broadband increase above baseline. So we can see the difference for how these different signals are driven by different images. And then if we can extract that from many images, we can start making models and predict what type of images drive each of these signals. And so if we do that, for example, if we look at one electrode, we can see that this is baseline in black. This is the frequency spectrum of power on the y-axis. Black is baseline. Red is when the subject looked at a grading stimulus. Blue is when the subject looked at a noise pattern. And we have to note here that this is a gamma oscillation. It's defined by a peak frequency, bandwidth, and power, whereas we have to decompose the spectrum into this narrowband oscillation because it sits on top of a broadband increase in power that spans many different frequencies. And because both things happen at the same time, when we decompose those, we can start comparing these two different signals to the fMRI bold signal and see what we can learn from each of these measurements about the visual input. So we showed 3 patients, 86 different stimuli that varied across space and with orientation. Look, for example, these are squiggly line patterns, gradings of different orientations, the number of orientations, so grading, plaid, different contrast, sparseness, coherence, and noise. And this is a typical fMRI response that was published before by Kendrick Kay. And they saw that the bold response for these stimuli in a particular voxel in V1 increased for certain stimuli on the right, stimuli on the top, and then had an overall smaller response to grading stimuli, and also increased for contrast. So we see certain stimuli driving the response larger compared to others. When we compare that to an ECOG broadband response to an electrode that is closely located to these voxels that are measured in a different patient, but in a similar cortical area, different subjects, comparable cortical areas, we can see that the ECOG broadband response is driven by very similar features, and that also the response to these types of squiggly lines is larger than the response to gradings. And that, for example, the number of orientations, these responses are larger or on par with the grading patterns, and then it increases for contrast. And because these 2 responses are driven by comparable visual features, we can build on image computable models developed for fMRI and basically simulate the network to predict a response. And so we filter the images with Gabor patches, calculate contrast energy, integrate across a population receptive field, and predict a certain response, and explain about 80% of the variance in the ECOG broadband signal. So which indicates that in terms of fMRI and broadband, it overall encodes similar features. So the visual input drives each of these in a similar manner. Whereas the ECOG gamma response, when we extract this gamma oscillation very carefully, which was the narrow band peak above the broadband increase, we can see that it's not really driven by these stimuli in this electrode and only by these grading patterns, and also has a larger response for increase in contrast. And so it seems to encode orientation variance, which is basically how many orientations you have. If you only have a few oriented gradings, that's what seems to drive these oscillations. And we can build a model with those features that is sensitive to those features in the receptive field and explain about 75% of the variance. So we have different stimuli drive the visual cortex, the inputs in different ways, and we can use these different signals to learn different things about the system, as long as we carefully characterize our power spectrum. And this stimulus specificity for gamma oscillations has also been published, for example, by Brett Foster's group in detail. He's with some more extensions across color. And so the question is not whether we see any narrowband gamma for one particular stimulus, but how we can explain observed variability and whether we can make models that predict this. And these image computable models make quantitative, testable, and interpretable predictions about the type of stimuli that drive this response. And within the receptive field, orientation variance and contrast in the population receptive field predict gamma amplitude, which are interestingly related to gain control or suppression of neural circuits rather than actual communication between regions. So what have we learned? We switched from a typical approach where we compare the signals directly and had a problem with that in photosensitive epilepsy to this approach where we think about what human brain measurements encode. And so if we think about this in terms of photosensitive epilepsy, we saw what type of features drive gamma oscillations. And if we now compare those type of features, such as increasing noise, reduced in our model and in our data, the amount of the power of the gamma oscillations, there have been a lot of studies that also showed that these types of features, this adding noise to a stimulus, also reduces interictal activity or epileptiform responses in EEG when you reduce noise in an image. And so we can do this type of comparison for many different types of stimuli, like, for example, the size of a grating increases the amount of that power in gamma oscillations, and also increases the number of epileptiform responses that a patient will have with photosensitive epilepsy. And we can see that across these features, contrast orientation, etc., there's a very similar, comparable effect that these visual input have on gamma oscillations and epileptiform responses in photosensitive epilepsy. Neuronal population firing rates or fMRI responses do not have that comparison. So when we take this approach about what human brain measurements encode, we see that not all stimuli that increase neuronal firing of or fMRI bolt signals elicit seizures. And it's particularly those stimuli that induce narrowband gamma oscillations that elicit seizures. And so we can, for example, propose this a hypothesis that gamma oscillations and epileptiform responses in photosensitive epilepsy are driven by the same circuit. And it's particularly this circuit that is affected in photosensitive epilepsy. So if, for example, if gamma oscillations are related to this type of gain control that breaks down, that may make patients more likely to have these seizures. But that's speculation, of course. We don't know that that's what's going on. But we have a model, so we can implement that also on, like, for example, an iPhone app, and give it give a number for how likely it is that you're going to have like a large gamma oscillation when you look around a room. Maybe this is helpful for identifying features that can also, potentially provoke seizures. And when you look at this around, patients with migraine also often feel some photosensitivity, and they particularly report that escalators, as you can see here, can make them dizzy. So it's interesting to kind of see that, compare these stories from some of the patients to what like a model like this would predict. You can see that there's also like these types of, it's not very common. We really had to search for some of these patterns outdoors in particular. So when we integrate human brain measurements, we can better understand maybe some of these diseases and really compare what type of signals drive them. And so when we saw these type of features, what we wanted to understand is also how can we treat some of these patients? Is it just like, can we use electrical stimulation to influence the visual pathways? And how can we do this? And one of the things that we noticed is that when we think about electrical stimulation to influence visual pathways, if we go back to the visual literature, there's been there's many years of very solid research in vision, measuring EEG signals and quantifying what every different response will look like for different types of inputs, and also with development. And we see that like visual stimulation and visual evoked potentials are extensively studied. And for example, flash evoked potentials, where when people wear an EEG, or the kids wear an EEG cap, but when they see a flash, they know exactly what a typical response should look like from preterm to term. So all these types of features, we know this for visual stimulation. When we put an electrode in a person's brain in one area and we record in another area, we don't necessarily know what this should look like across age or across different regions. And so these evoked potential waveforms, they inform about typical and atypical visual function. And we want to start understanding how that works in the visual field. Like we first want to do before we're starting to understand how we can use these measurements to maybe help patients, we want to have the basics, like cover some of these basics, develop approaches that can do this in different systems. So when we think about how electrical stimulation influences visual pathways, I would like to take a step back first and understand how brain stimulation evoked potentials, how those waveforms inform about like these network pathways, for example, and the nodes. And then I would like to show you an example of how we're using pulvinar stimulation to influence select cortical areas in epilepsy. So we call this brain stimulation evoked potentials. Originally, these were called single pulse, like cortical evoked potentials, because people were typically stimulating cortex here and cortex there with ECOG electrodes on the surface. But since we're also using white matter stimulation, pulvinar stimulation, we can't really use the CCP terminology anymore. And so we're starting to call these brain stimulation evoked potentials. Hopefully that'll capture most of the findings for now. And so recently we've seen the shift from these cortical surface recordings to stereo EEG. And for example, we stimulate an electrode pair here with a brief bipolar pulse, and then we can measure the evoked potentials in directly or indirectly connected areas that happen like after the stimulation happens. And you can see here that after stimulation, there's first this brief artifact for about, that happens for about 3 milliseconds after the evoked response. And then we see the evoked response. Depends on your recording setup, how long the artifact is. And then we can record, these are across trials. You can see that across trials, you can see that this electrode responds to a particular type of stimulation in general, but with varying amplitude. And so we can average pulses in one side of per stimulation pair. And one of the first features we wanted to look at is more if we think about how we stimulate one side, if we think about the spreads may spread through the white matter, and it would end up after a certain amount of time in a different area, what a typical latency would be, because we actually don't know this in the human brain. There have been different reports reporting things from 10 milliseconds to 20 milliseconds to even 40 milliseconds. And when we think about that, we have to think about if we have a network in a human brain, what's a typical conduction delay in these visual pathways? What should we expect? And we actually couldn't know this directly from, and this is the conduction delay is of course driven by the axon diameter between these areas and the myelin between these areas. And so when you think about this is on the left here, you see across years, the myelin fiber length density in humans. And here on the right, you can see this for a chimpanzee. And what you see is that chimpanzees do, basically plateau after about 10 years of age, whereas after 20 years in a human brain, you have this additional stage of myelin development. And so if we want to understand how fast things develop in human brains, we actually have to do these measurements. We can't just extrapolate from an animal experiment, except if we knew all the axon diameters in a particular bundle. So we thought we could just at least do some measurements and see what we find here. And here we're looking at the superior longitudinal fasciculus. These are electrode locations across 74 patients that had electrode positions on the areas that could have endpoints of the SLF in frontal and parietal regions. And let's look at two subjects first, and we can see that on the cortical surface, one of the most common features that we see in these responses is a negative deflection after stimulating another site. And the dashed line is the response in a 38-year-old, whereas the solid line is the response in a 4-year-old. And you can see that there is a twofold increase from the adult to the child. This is about 25 to 30 milliseconds, where this is 50 milliseconds. This is not a small difference. If we were to do, and so one of the questions we wanted to know in this case, like, is this the same response? Is there something different happening in the child? What is the difference here? So we did this we calculated these peaks across the patients that had this type of coverage. And what we saw and we sort them according to their age from 4 to 51 years old. And what we see, for example, across the temporal parietal Aslan tract or across the SLF, we also looked at the arcuate fasciculus and at the central to frontal part of the SLF. We see this decrease in latency with development, and it was extremely robust across patients. I mean, you can see that there's variability as well, but across patients overall, the adults are about twice as fast as the kids. So we can still be happy when we're 40. And what you see also is that if we correct for the brain size and we co-register an atlas of fibers to an individual patient and estimate the signal propagation speed is that it increases from about 2 to 4 meters per second. And to give you an idea of like how fast that is, that means that we're, at about a walking speed as a child, and at a biking speed when we are about 30 to 40 years old. And so what we've just learned about is how these networks change or can change across patients. So we have to take these patients' differences into account. And so we've learned about how these pathways can influence how we stimulate a ventral area, for example, a visual ventral area. And that if we're working with a child, we see a lot of kids in the intracranial, for the intracranial measurements, that those responses may be slower compared to an adult. And that's normal. That's not something that's abnormal. And so when we just looked at these, like how we have to take this, conduction delay into account, and we can look at these early responses. But we also can think about the measurement side, so the node in the network. And when we look at these type of responses with stereo EEG, what we actually find is that there's much more to discover than this early negative response on the surface that we would see on the surface. There's many different waveforms, many different later responses. And so what can we learn about those things? And when we think about that, in a convergent paradigm, so input from many areas into a single area, we can think about that as different inputs. If we look at a single electrode, if an input arrives in a deeper layer or a superficial layer, we would expect that the response actually has a different waveform. And we know that this is common in visual pathways, where, for example, Velleman and Van Essen already showed that ascending feed forward or feedback or lateral projections have different inputs. This is what happens in the input layer to different laminar structures. So even if you measure with one electrode on the cortical surface, because the fields are different, you would expect different response shapes. So we wanted to test whether we could see this and whether we could see this in our measurements. Do different types of inputs cluster in terms of different responses? And a good way to test that is also in the ventral temporal cortex, because it receives inputs from visual areas, from memory circuits, from emotional areas. We know that this is this like high input area, and we had electrodes there in several patients. For example, here there's the collateral sulcus. These electrodes were a little bit deeper. And so when we look at one of these electrodes, and this is first we developed this algorithm and then to look at these different response shape in an ECOG patient with ECOG recordings. So we stimulate here at the green site and measure here in this red side. And we see that after we stimulate, we see these types of positive responses after stimulating the green site. However, when we stimulate this purple site, we see a very different response, and we see these rapid negative responses after the onset of stimulation. And when we look at all these, so what we're going to do is we're going to look at the measurements in a single site, and we're going to just plot the responses throughout the brain, whether we see a pattern here. And I don't know if this is visible at all, but let me cluster it for you. What we see is that if we plot in these red traces what the response in this single site here, the single measurement site looks like, we see that these green areas show this positive response. So they're spatially co-localized. And these yellow sites trigger this negative response that's slightly different, also negative response for this purple site, but that also has a positive waveform afterwards. So we see these clusters emerge visually with different stimulation. So when we're stimulating these green areas, we evoke a different response waveform in this red side compared to when we stimulate these yellow areas. So we can drive this area in different ways. And we wondered how we could identify these algorithmically. And standard approaches to clustering didn't really apply. And so we developed this method that we call basis profile curve identification. We wanted a set of these types of curves. How does a stimulation area drive a second area with canonical responses that are present in the cortical evoked potentials or brain stimulation evoked potentials? We wanted these two responses to be weighted by the significance and like the amplitude of each stimulation site. We wanted a non-negativity constraint because if you're measuring in a single electrode, a positive and a negative response imply fundamentally different inputs. It means that there's a different field around that electrode, different electric field. And we did not want an orthogonality constraint as some shapes could be shared. And so we based this method on cross projections between trials. So for each cross projections across a single trial responses. And so when we project one trial into another, the cross projection is going to be large when it's very similar. It's going to be small when it's very different. And so when we do this across all trials, we get this array of cross projections that we can then basically see whether it's larger than 0 or close to 0. And we can put these amplitudes, for example, we have 41 stimulation sites. We can do the 40 stimulation sites by 40 stimulation sites and see that some are similar and some are very different. And we can use non-negative matrix factorization to reduce this matrix to a set of clusters, like 3 different cluster types of responses that are very similar. And so we can see that in this case, one cluster describes this negative response, another one that has a different waveform, and then this positive response. The MATLAB code is available online, and you can also find the data in BIDS, and feel free to reach out with any questions about this. And so when we see these different responses clustered, we can also plot that back on the brain surface. And we see that the weights from this green weight that were driven in this electrode, this is the measurement site, were driven by these green sites, etc., for the purple and the yellow sites. And these match what we drew on the brain earlier when we just looked visually, that we saw these negative responses here and these positive responses there. And so it's a crucial question to ask now whether these characteristics are similar across subjects. Are we just seeing this? Can we do this on a single patient, or can we actually extract more general principles about how different areas drive the ventral stream, for example? And Harvey Wong, who's an MD PhD student who just graduated in the lab, wanted to look at the collateral sulcus. So he looked at across subjects an electrode in the collateral sulcus that was on the gray-white matter border. And for example, this was one of the electrodes. And when he used these basis profile curves across many other stimulated sites, he found that there were 3 specific waveforms. And when we looked at the sites that were stimulated for this waveform, he found that the hippocampus drove this particular early negative strong response, whereas the amygdala elicited a very different response in this area. And the insula and other cortical areas elicited that response. When we plot these electrodes on a cross section, you can also see that these blue electrodes, that their stimulation elicited this response, were in the hippocampus, whereas the cyan electrodes are more located in the amygdala. And when we inflate the cortical surface, you can better see that these other electrodes that elicited the positive response were in the IPS and the MTG in this case. And so this was in one subject, and now we go across subjects. So we did this across 5 subjects and got all these basis profile curves and put that in a principal component analysis. And then we got still very similar waveforms. We got this blue waveform, and that was generally driven by stimulating this blue site, the cyan, by stimulating the cyan site. And when we look at those areas and see what the anatomical location was of the position between the 2 electrodes, we see that this blue curve was across subjects driven by hippocampal stimulation, whereas this cyan curve was driven by more amygdala stimulation. And the green curve, mostly by lateral temporal stimulation. And this last waveform was more of a mix. But what this shows is that a shared shape We can see shared shapes that come from stimulating similar structures across patients. And all these patients, we included these patients because they didn't have epilepsy in this network. And now we can start seeing how this, for example, differs when patients have epilepsy in one of these, epilepsy affects one of these areas. The seizure onset zones were all in the frontal lobe or other areas. And so in addition to these kind of evoked responses, one of the things I would briefly like to address is that there's also spectral changes. So we can really drive the neuronal activity in these areas. And that is also not specific for all stimulation types. Specifically for this first hippocampal stimulation, we see this large suppression in the power spectrum from about around 20 to about 150 hertz at least. And from about 100 to 300 milliseconds after stimulus onset. And this was similarly seen for amygdala stimulation, but not for the other types of clusters of electrodes. So we can see the suppression in broadband activity after a brief increase in activity. And so what have we seen now? In terms of paths, we see that responses change across in adults compared to kids. Timescales change. Developmental trajectories are comparable to different diffusion MRI measurements and are likely driven by microstructural changes like myelin and axon diameter. And the timescales of neural signal propagation change across the lifespan. In the nodes in the network, we can see that when we stimulate other areas, is that ventral temporal cortex shows input-specific response waveforms. Stimulating different areas elicits a different waveform. This is like saying it's in the visual world, this was as basic as like a different, a flash versus a grading versus a moving dot pattern drives a different response. We need to understand this not just in the visual pathways, but in any of the other regions that, for example, Dr. Halpern is looking at in all these responses. There's a lot to be discovered, but if we do that systematically, we can start linking this to particular circuits and understanding how different circuits influence these waveforms. And even when we looked in the subject, not in the collateral sulcus, but when we move one gyrus over to the fusiform pathway, the responses, we show that the responses also differ. So it's really network and location specific. We have to be very detailed when we look at these different response waveforms. In addition, we find that there's sometimes there's interesting spectral power changes, and that hippocampal stimulation induce these increases following by these decreases in broadband power. And we think that these may be potentially related to short excitatory responses followed by longer lasting inhibition, because previous studies, for example, by Logothetis, showed that LGN stimulation elicits this type of response pattern in V1. And so we can start to understand these types of responses in these visual pathways and map these visual pathways out. And so the last question that I would like to address is whether pulvinar stimulation can we actually use this type of electrical stimulation to influence these types of epilepsies in these visual pathways? And there are several case series and case reports right now that actually show that pulvinar stimulation can benefit posterior quadrant epilepsies. And so there's a list of actually 5 case series that have shown that that benefits. But the pulvinar is a big area. It's a large nucleus. I mean, what does pulvinar stimulation mean? What are they stimulating? And we can see from this diagram from Mike Arcaro's work that the connectivity for different pulvinar subregions is also expected to be different. So for example, here, this is the medial pulvinar. The ventral part of the medial pulvinar projects to middle temporal areas, whereas the dorsal part projects to parietal and frontal areas. Whereas the lateral pulvinar, and mostly the ventral part, projects to early visual and extrastriate areas. And so there's these different outputs from the pulvinar, and we can test now with electrical stimulation, with a single pulse electrical stimulation, to see whether that also, when you stimulate these areas, where there's also different influences on these different regions. So we had 12 subjects who had electrodes with contacts that were close to the pulvinar. They targeted either the CM or the pulvinar nucleus. And Jordan, who is a research assistant actually in the lab, organized all these different patients and clustered them and looked at the responses when we were stimulating with a single pulse through these different electrode pairs. And we specifically looked at more of the lateral pulvinar stimulation, ventral pulvinar stimulation, and dorsal pulvinar stimulation, to see if we would find a similar connectivity pattern as the anatomical connectivity pattern that was shown before using diffusion MRI, where the lateral pulvinar would influence early visual pathways, this ventral medial pulvinar would drive temporal areas, and the dorsal medial pulvinar would drive more parietal regions. And in order to do that, quantitatively, we had to quantify all these different waveforms of responses and see, independent of what type of response we would get, whether the response was significant or not. And we call this canonical response parameterization. And we again use these cross-trial projections to see if the response was significant or not. But in some cases, the pulvinar may elicit like a long response that lasts like, half a second. Whereas in another case, it might have a very brief but reliable output. So how do we put these different response types on the same, how do we quantify that in one number? And so when we use cross projections between the trials, they are again large when it's reliable and small when it's not reliable. We can do that with increasing numbers across time. And you can see that these cross projection magnitudes increase until the response becomes less reliable, until the response is done, it becomes noisy, and then it declines. And so we can use this type of response parameterization to get to time, just a time where there's a significant response, and only use that for our statistical analysis. When we have that response time, which we call tau, we can average or use a PCA to find the canonical response in the time and predict that into every individual trial to see how reliable that response is. So that gives us several ways to parameterize every trial or every response that we see, despite these arbitrary shapes. And in this case, we use the explained variance as a measure to measure this kind of effective connectivity strength. How strong is the pulvinar driving all these different regions? And I'm first going to show you like a smooth group map of what this looks like. And then I'll show you some of the individual results that back this finding up. So when we look at pulvinar stimulation and we look at the lateral pulvinar stimulation, medial pulvinar stimulation, and dorsal medial pulvinar stimulation, we see that this is the average, the smooth kind of strength of connectivity map across the subjects. So this is a smooth map controlled for the number of electrodes in a certain spot, controlled for the density. But what you see is that when we look at these occipital areas, is that it's specifically the lateral pulvinar that drives a stronger response there, a more reliable response there, compared to these other types of medial pulvinar stimulation. Now let's look at some of these, and this is exactly what we would have expected because the lateral pulvinar has the strongest connectivity to extrastriate areas. When we look at the lateral, we take the lateral view, we can see these more middle temporal areas. We see that the ventral medial pulvinar drives those areas the strongest. There's also, this is like in these patients, they have some overlap of the electrodes. So it's not an all or nothing, but you can see that this is the strongest out of all of the three of them. And that this maybe drives more of the occipital areas and this also the anterior sites. When we take a posterior view, so we're looking at the back of the brain here, and we're peeking there into the interparietal sulcus, we can see that that's largely driven, or more strongly driven, and more strongly influenced by the dorsal medial pulvinar. And so in individual patients, and so those are the group averages, you can see that we can selectively, there's a selective influence across some areas with certain pulvinar subregions. And you can see this also in the individual subject. For example, in this individual subject, you can see that the electrodes go here from more of the medial ventral pulvinar site to more of the lateral, a little bit more dorsal pulvinar site. And you can see that when we stimulate the medial pulvinar, on these early visual areas, we do not see a strong response. Whereas we do see a strong and reliable response when we stimulate the lateral pulvinar. And this is just switching from electrode number 1, 2 to electrode number 2, 3. And these electrodes are just a couple of millimeters separated. And we see at the same time in the same patient on the lateral temporal surface, we see that when we stimulate this medial site, we see these strong outputs to lateral temporal areas. Not all electrodes, but definitely overall we see the strong outputs. Whereas when we shift over to the lateral pulvinar, we do not see those outputs. So this pattern matches extremely well with what the diffusion imaging showed before. And we similarly see that across the dorsal pulvinar, we just in individual subjects, we also see that we drive the IPS stronger, whereas this ventral, also in individual subject, the ventral pulvinar just drives these lateral temporal areas most strongly. And the question is now is like, if we have such specific outputs, can we then use this in epilepsy patient to help patients with epilepsy? And we do these single pulse stimulations generally when patients are back on their medication, but before they are back on their medication, they sometimes receive a trial stimulation. These electrodes are implanted in the pulvinar because there have been these clinical trials, and the neurologists want to test whether stimulating the pulvinar can actually help those patients. Like if a resection is not possible, they don't want to cause a hemianopia in these patients. They want to see whether stimulating pulvinar can reduce these interictal discharges. And so Greg Worrell and Nick Gregg are two of the neurologists that I closely work with on these topics. And so what happens in these patients is that we often first do this single pulse stimulation to map the thalamocortical connectivity, the pulvinar to cortex connectivity. And then also there's a baseline, there's a lot of baseline recordings where there's an interictal epileptiform discharge rate that's calculated. And then they stimulate with a high frequency deep brain stimulation, like 145 hertz in these pulvinar electrodes. And then they again see what the interictal discharge rate is across that time when they're stimulating. So we first map the network, and then they do these types of measurements. So what we see here is that during the pulvinar stimulation, this is the seizure network activity with these red dots marking the interictal epileptiform discharges, is that during these trials of 135 hertz here in yellow, that there's a large reduction in these interictal discharges, in specifically the seizure network, in more of the lateral temporal, more occipital areas that was affected in this patient. And so we can use maybe this thalamocortical connectivity to understand whether this seizure reduction rate is specific for the network that is actually influenced by the stimulation. And there's different ways in which we could imagine that. If there's no relation between this thalamocortical or pulvinocortical connectivity and the change in interictal discharge rate, we would expect that in all the patients, we would just expect that the interictal discharge reduction in an electrode in the seizure network is independent of like whether it was connected to the stimulated site or not. We could also expect, like if there's a one-to-one relation between this thalamocortical activity and the amount of interictal reduction you would see, you would expect that there's a negative correlation between the two, but also that in every individual patient, there's a relation between these. If we would expect a per network effect, so you see that there might be a strong input to this electrode, weaker input to this electrode, but overall the interictal discharge rates are reduced in all the electrodes that have some input and not in other cases. We would expect a per network effect across patients. When we do this calculation, so we do this baseline thalamocortical connectivity, stimulating the pulvinar, looking at those different in one patient, but other patients had, for example, ANT stimulation and CM stimulation, looking at the connectivity patterns and seeing whether there was a reduction in interictal discharges. We see that we find evidence for the per network effects. So overall, across patients, the patients that had large thalamocortical connectivity also had reduced interictal discharges in the connected network. But if the seizures come from a different area than what's connected to that part of the thalamus that's being stimulated, there's not going to be an effective treatment. So this mapping these types of connectivity is extremely important for these types of trials. So to summarize, I first showed how we can integrate these human measurements, brain measurements, to better understand photosensitivity. And they provide complementary information about neural processing. And we can make image computable models to make quantitative and testable prediction on the type of image features that are encoded in different brain signals. And if we do that type of careful distinguishing of all these different signals, we can come up with hypotheses like that gamma oscillation epileptiform responses are driven by the same circuitry. And then we can start to test whether we can use electrical stimulation to influence these visual pathways and maybe help some of these patients that have particular disorders in the visual areas. And we can see that electrical stimulation-driven neural signal propagation varies with anatomical properties of the path and the nodes in the network, such as myelin, what type of projection we have, also probably whether we'll have direct or indirect projections, etc. And we can use pulvinar stimulation to influence select cortical areas. And we have to be very careful about where exactly these electrodes are placed in the pulvinar. Because electrical stimulation-based connectivity predicts the impact of the brain stimulation on epileptiform activity. And for all the published manuscripts, not the ones that are on bioRxiv, but for all the ones that have been fully published, there is accompanying de-identified data and code shared on with the manuscripts, such that the main figures can be reproduced. And please contact me or post a GitHub issue if you have any concerns. And the data, we generally try to share those in bids because it helps loading them. And so I would like to acknowledge the lab in Rochester and also my wonderful collaborators and my funding, of course. And I would like to thank you for your attention.
Yeah, so this was super interesting. I thought the part about image computable models to predict epileptic activity was really interesting. I wonder if subjective experience of perception has anything to do with it. For example, you could imagine two overlaid images, one is a grading and one is not, if you're paying attention to the grading or not. Or if it's only visible when you fuse the input to both eyes, you know, some kind of like free fusing. Do you think that that could have an effect on how those types of stimuli can produce epileptic activity?
So this is actually an interesting question, because when patients, for example, with one of the recommendations on the network against, for some of the epilepsies websites, is that, and also from the neurologist, is to avoid looking at these gradings, just avoid looking at those patterns. But if you're in a situation where you're looking at those patterns, close one eye.
Interesting.
Closing both eyes, especially if there's a flashing light, sometimes makes the flashes even more provocative. So it's interesting that they are generally recommended to close one eye to disrupt this activity rather than,
Cool.
close
Interesting.
both eyes. So I'm not sure how necessarily how that works basically between the ocular dominance columns. But that's one of the recommendations that they clinically give. So there's definitely some level of visual input dependent experience that you can control.
Cool.
Yes.
Awesome talk. Thanks, Dora. So many exciting projects. I want to jump to the pulvinar because that's my favorite thing. I'm curious if you had, and maybe I missed this in your data, like what you think the pulvinar is doing as opposed to like other areas that might be providing feedback to the target region that has the source of the epileptic seizures. Like, is there some, pulvinar things that have some specialized circuitry, maybe playing a really important role in facilitating cortical communication? Something about that particular with the pulvinar versus, it's just sending some, signals to the area.
So, so we're working on more of the interpretation in terms of our cortical measurements. So, but in general, a couple of things based on the stimulation outputs. Just when you, for the thalamic stimulation, the the reason why the neurologists often choose these thalamic targets, because stimulating those areas can generate widespread inhibition or widespread suppression of cortical activity. That's what they think is going on. Whether that's actually, like, whether we see that in our measurements, I'm not sure. Because it would be, if we're interrupting this circuit, are we causing an interruption of the circuit in the pulvinar, or are we adding noise that would have a different effect? But it generally thought that stimulation there has a suppressive effect on the cortex. Whether that circuit is actually suppressing the cortex or not, I'm not sure. So there's a difference between what the stimulation does to the circuit and that does on the cortex compared to what the circuit directly would do on the cortex. So that's one of the things. The other reason why the pulvinar is generally, or like these subcortical structures like the thalamus are generally more effective than another cortical structure, is because they have widespread outputs. So even though they are selective for certain regions, they're relatively widespread across they're not just driving one electrode on the middle temporal gyrus, they're driving like 10 electrodes. They're driving like a bunch of the electrodes on early visual area. So it's easier to cause widespread disturbances in the network. And then when you do this across longer times, this is also the effects that we see here are occurring, like the suppressive effect over longer times occurs after about an hour and a half of active stimulation. So it's generally delivered intermittently. So if you do, like, say you stimulate for 1 second on, 2 seconds off, you would have to stimulate for 4.5 hours to obtain an hour and a half of active stimulation. So these suppressive effects that we see of the pulvinar stimulation are specifically observed after this time. We don't see them after only in less than an hour and a half of active stimulation. We see much more variable effects. So it's a long-term effects that we're thinking about in this case. So this long-term stimulation of the pulvinar has these effects. The short-term effects, however, in function, I'm very interested in that, but I just am not sure about yet. Yeah. Yes.
Dora, I've always wanted to ask you, we use your methods obviously to help with the mental health, understanding mental health related networks. And the electrodes we use are temporary. But we do all of our connectivity testing with those electrodes. But then we take them out and we place, more permanently implanted clinical electrodes, which are often very similar, but not identical. And I was just curious, how you would manage that, because it is a little bit of a confound in terms of interpreting what the chronic stimulation effects are versus the acute effects, which you are doing, immediately with the same electrode. The chronic effects are being done with slightly different electrodes. And, do you think is there a way to sort of make some assumptions that are reasonable?
Well, so one of the reasons why we're doing this type of stimulation across different patients, right? Or different humans that have dramatically different types of, not dramatically, but have very different types of anatomy. So if we can generalize across two or 5 patients, as we showed in the ventral temporal study, that had, and the hippocampal stimulation just has this particular waveform in the collateral sulcus across the patients. Then across other patients with similar electrodes, I would probably expect something different. It depends on what the order of magnitude is where this changes. If you go from macro to micro electrodes, like how small does the electrode get to drive a similar amount of cortical tissue? I mean, that's kind of what we don't know yet, is what the sizes are across which these signals are distributed, for example. We can get some idea from between different electrodes, of course. And we know that they can change on a 0.5 millimeter type of, spacing, or like, 1 millimeter spacing. You can see different electrodes. If you have laminar electrodes, you would probably get much more detail compared to, I think 2 or 3 electrodes span that area. So it depends on what type of detail you're looking for. So that's in terms of the measurements. I think there's large differences. In terms of the stimulation, that would depend on how much cortical tissue you're driving. If you're putting in a smaller electrode, you can have a larger volume of tissue activation, probably more comparable to a smaller volume of tissue activation. If you have a larger electrode, I mean, I don't know if you can exactly match the sizes, but it would probably be comparable in terms of how you would drive the network, as long as it's still orders of millimeters rather than you go to micrometers. if you start to jump orders of magnitude, it would probably be very, very different.
Yeah, I think speaking at a high level about the differences, I suspect they're about 0.5 to 1 millimeter different. Either because of surgical placement or because the spacing of the electrodes is slightly different or the width of the contacts is slightly different. Probably plus or minus 1 millimeter is what we're looking at.
So when we look at with the pulvinar stimulation, for example, if we look at electrode 1, 2, and 2, 3, and 3, 4, electrode 1, 2, and 2, 3 can sometimes be very, very different, but then you can also change the amount of current that's delivered to electrode 1 and 2 and make it, I mean, I guess that's where the state of the art neural stimulation comes in, what you want to achieve. In the cortex, this can have even larger effects because electrodes 1 and 2 can be in the gray matter, 2 and 3, if 2 is on the border between the gray and the white matter, 2 and 3 can be crossing into the white matter. And if you have 3 active, you can have a dramatically different response. So in that case, I would think that those 2 or 3 millimeters would, maybe 1 millimeter, but 2 or 3 millimeters would have we've seen that can have a dramatic effect. And I probably should put that in the slide just to emphasize that type of specificity at some point. Yeah. Thank you.
All right, every millimeter counts. So
That's right.
let's thank Dora again for a very